Hi everyone, I'm Fairlie. I'm a fourth year in the Mortazavi lab, and today I'm going to be giving you the most boring talk of the day, but hopefully it will give you some foundations and understanding on how you can go from raw sequencing data to actually something that makes pretty UMaps and pictures down the line. So unfortunately, due to time constraints, we aren't going to make any pretty graphs out of this data today, but we'll get you running some pre-processing software that you can use on your raw sequencing data, raw single cell RNA sequencing data down the line. And some of these principles that I'll be introducing are applicable to other sort of genomics data types. So even if you're not going to be doing single cell RNA-seq, if you're going to do ATAC-seq or RNA-seq bulk, these principles are things that are ubiquitous throughout genomics. So even if it's boring, and I apologize for that, it'll be applicable. And also, as with the last session, if you have any questions, please let me know. I haven't presented this stuff before, so I want to make sure it's clear. All right, so let's get right into it. First of all, what does raw sequencing data look like? So on the right here, I have an example of what two reads look like in FASTQ format. And this is what the reads look like, usually directly after they come off the sequencer. Each of these entries consists of 4 different lines, 4 lines per each read. So what you're seeing on the right here are actually 2 different reads. And so as the slide indicates, each of these lines has a different piece of information. The first line in blue is the read ID. It always starts with an @ sign, and it is like a unique identifier for basically some metadata about the read, including what platform it came from. So like if it came off an Illumina sequencer, as well as perhaps where in the flow cell the read came from. So it's not particularly useful other than the fact that it is unique within the FASTQ. Oh, and sorry, I don't know if I mentioned that, but this is FASTQ format. So when you get a read off of a sequencer, it'll be in a file that's like .fq or .fastq. The second line here in red is the actual nucleotide sequence. So you'll recognize all of those letters in that red sequence over there. They're AGs, Ts, and Cs. They're the genetic code. That's exactly what you're going to be sequencing when you're actually sequencing your data. The third line is not important and has only ever been explained to me as a placeholder. So don't worry about it. I don't know why it exists. And the final line, the fourth line in green, is the quality score for the read. So basically there is kind of like a score key that is used in FASTQs and in sequencing to indicate how high quality the sequencer was able, or how high confidence the sequencer was able to determine each base in the read. So you'll notice that each of the red sequences is the same length as the green sequences. So there's a 1-to-1 mapping between the green sequence and the red sequence. And if you look up pH Fred, like Fred with a pH score, you can learn more about how that score is computed, but it's typically used for QC down the line. Okay, so you have your raw sequencing data, but it doesn't tell you a whole lot about what you're actually looking into. And so this talk is going to be guided by 3 main questions, which we're going to go through step by step. The first of which is, how can we determine what cell each read comes from? So obviously, if we have single cell data, each read is going to have a cell of origin that it came from, but that is not immediately clear from the sequences that we see here. Another question is, how can we determine where each read comes from within the genome? So even if we know what cell the read comes from, we still don't have an idea about what gene is being expressed that underlies that read. And finally, we want to be able to take these reads and turn them into an expression matrix or a counts matrix. We want to determine how highly expressed each gene is. And so I'm going to go through these step by step. And again, please feel free to stop me if anything is confusing at any point in time. Okay, so the first question we're going to answer is how can we determine what cell each read comes from? And the answer is actually pretty simple. Cell barcodes are going to be sequences in the red sequence, the second line of the FASTQ, for each read here. And typically in short read, this is very easy to grab the cell barcodes because your library preparation is going to dictate where exactly in the read the barcode comes from. And this is a consequence of Illumina short read platforms actually having a fixed read length. So every single read that comes off of the sequencer is going to be a specific number of nucleotides long, say 50. And this is because you literally run the sequencer for like 50 cycles, and that is the number of nucleotides you incorporate. So using this principle, basically, if you know where the barcodes are being ligated to your constructs, so say in this example, we have barcode sequences that are 8 base pairs long, and we know that they're going to be at the end of the read. We can simply just take the last 8 base pairs of each read and call that the cell barcode. And in this way, each unique combination of 8 nucleotides that's seen at the end of each read indicates the cell of origin. So every time you see the sequence that's in that first barcode on a different read, that's going to indicate that those reads came from the same cell. So it's pretty simple. Okay, as an aside, I'm going to talk about an additional type of nucleotide tag that's often put onto the different molecules in these experiments that Liz actually mentioned in her talk pretty briefly, which, oh, sorry, I forgot about this slide. Different single cell library preparation methods have different barcoding strategies. So these will dictate how the barcodes are actually extracted from the reads in the different software that you will run. So for example, 10x on the left has 26 base pair barcodes, and SplitSeq on the right, which Liz also talked about, uses combinatorial barcoding, where you actually have 3 different 8 base pair long barcodes. And so this is an important consideration when trying to determine the cell of origin for each of your reads. This is also a process called demultiplexing. Okay, so I'm now going to talk about unique molecular identifiers, which Liz mentioned in her talk as well, which are other sequence-based like additions to these reads that help you control for, bioinformatically control for PCR duplication. Yeah? Yeah, so as with anything in biology, there are errors. So there are a lot of considerations that actually go into designing these barcodes. It's much like primer design or something like that. You need to make sure that they're going to be like distinguishable from the actual gene. So people will like make sure that it doesn't overlap with a region of the genome so that you don't accidentally think that that's where it came from as opposed to from the barcode. Other methods will actually correct for barcodes. So say you have like a set of 10 barcodes that are just literally the oligos that are included in your sequencing kit. If you see something that is like close to one of those, but is not in that list, you might be able to correct it by checking how close it is in terms of its similarity to a barcode that's predefined. So those are some sort of strategies that are used to detect these barcodes. Yeah.
Will you always see the barcode at the end or in the middle sometimes? Or?
Yeah, so it depends on the library preparation strategy. Yeah. And I will also say that this is probably another place where those like quality scores come into play, because if you have like a low quality base in your barcode, that might indicate that it's not super trustworthy. So you might be able to correct based on that fact too, but I don't know if that's actually how the algorithms work under the hood. Yeah, what's up?
Do you ever sit down and like actually look through your tests?
Nope. I think it's good. I think it's good for people to know what they look like though.
Nope.
Yeah.
I was just curious.
Yeah.
I never found
Yeah, I mean, I don't know. I think it's good to like, if you just like look at the top of your file, just make sure it's the right file type at least. Make sure you're not trying to run it on like something complete gibberish.
Oh, actually, can I add something?
Yeah.
You don't need any specific tools to actually look at this data. In other words, it's human readable.
Yeah.
Like it's not.
Like you could open this up in a text editor, and this is exactly what it would look like right here. It might crash your computer because it's too big, but yeah, okay. Okay, moving on. Now I'm going to talk about UMIs, and they are also sequence-based features that you add to your molecules in order to control for PCR duplicates. So I'm going to kind of walk through what this actually means. Maybe you already know what these mean, and I apologize for that. But say you have these 2 transcripts. Oh my gosh, I'm going to use my mouse. So you have these two transcripts that are present in your data. You have transcript A, the blue one with 3 copies, and transcript B, the red one with also 3 copies. And essentially what you want to be able to pull out of this information is that there are 3 blue transcripts and there are 3 red transcripts. But of course, there are some experimental steps that go that happen in between what is actually in the cell and what you're actually sequencing. So during your cDNA synthesis, you're going to tag each of these molecules with a sequence that is the unique molecular identifier. And here they're indicated by different colors. So when you undergo PCR amplification of your molecules, there might be PCR bias, which causes a specific transcript to be amplified more than a different one. So in this case, you can see that the blue transcript actually tends to be over amplified compared to the red transcript. And this results in there being twice as many blue transcript reads as red transcript reads. But in order to correct for this, because if you just took the raw counts as is, you would say, okay, the blue transcript is 2-fold more highly expressed than the red transcript, which is of course not the actual case in the initial population of transcripts in the cell up here. So what you can just do is count up the number of reads that have this a distinct unique molecular identifier, or UMI. So here you have 3 different colors or sequences of UMI, and here you also have 3 different colors or sequences of UMIs. So the actual quantification information you'll recover using this kind of like collapsing strategy is you'll have 3 copies of the blue transcript and 3 copies of the red transcript. And so much in a similar way to the barcodes, these are also typically added to like the end of the read during library preparation. So if we have UMI sequences that are 5 base pairs long and they are immediately upstream of the barcode, this is where they will be in the sequence. Okay, any questions on that? Yeah.
Barcodes, then I'll go through, but how does it make sure that it's like, oh, that one
Oh, so you're just counting, at this point, you're counting how many transcripts have, or basically how many unique UMIs you see per transcript. So you see blue here, pink here, gray here, that's 3. And then you see green, fuchsia, and orange, so that's 3 also.
So the other part.
Yeah, there's some choices that are made on like which actual read to like keep compared to the others, but I'm not going to get into that. Yeah, what's up?
So I think Liz mentioned that, let's see, there are other technologies that don't have UMI.
So SplitSeq does use UMIs. I'm not sure if she misspoke or something, or if she was referring to a different technique or something. Yeah.
Like if it doesn't, how would it deal with that?
Yeah, I don't know, but it does. So yeah, what's up?
So let's say you have a doublet or a triplet in your little droplet. What would you see? Let's say we have just 2 cells.
Mm-hmm.
Right? And droplet, let's say
Yeah, so they would all have the same barcodes. Typically, what happens in doublets is you might actually see an increased number of unique UMIs per cell, because that just indicates that there was literally like just more starting molecules, because you have more than one cell in your droplet. So say you have 10 molecules in your cell, in each cell, and then you have a doublet that has 20 molecules. So you see 20 unique UMIs in that, where everything else is 10. That might be an indication that it's a doublet. So that's one of the ways that you can bioinformatically control for doublets. Yeah. So the UMIs exist separately from the barcodes, as far as I know. So those will still tag each unique molecule differently, but the barcodes are the same across the entire droplet. So those will be the same barcodes added to each molecule. Cool. Yeah, this is kind of like a tricky thing to think about, but it is important because PCR bias is real. I don't know. I think that most techniques include it as part of the library prep. So most techniques would already control for that. Yeah. I don't know if anybody else has any knowledge of that. Okay, cool. All right. So moving on, we've now answered the question of how we can determine which cell each read comes from, as well as our question of being able to control for PCR amplification bias. So the next question is, how can we determine where each read comes from within the genome? And we do this with something called mapping or read mapping. So one extra file that you need for this is something called the reference genome. So there are reference genomes that exist for a whole bunch of different organisms. Here's a tiny fraction of the organisms that you can download reference genomes for that are available on the UC Santa Cruz genome browser. And these are files, text files, much like FASTQs, that you can open and look at if you want to crash your computer, that are in a format called FASTA. And this just consists of a header line, as you can see right here, that has a caret in front of it, followed by the chromosome name. So in humans, this would be chromosome 1 through 23, and then chromosomes X and Y, or 22, and then chromosomes X and Y. And then the second line is the sequence, the actual reference sequence of the chromosome. And of course, there is like a lot of variation within individuals of a specific organism. But at some point, decisions have to be made about like what the actual reference is going to be. So there's not enough variation that would complicate read mapping to a reference genome, yeah, for each individual. So we pick like someone to be the reference and are like, okay, that's the reference genome. That's what we're going to compare everything to.
So it's not an average.
I think it might be an individual. It might be an individual whose cell line they co-opted to reproduce ad infinitum, but I'm not sure. I don't think that it's an average because the average human does not exist. Yeah, I'd have to look into that though. That's a very interesting question. But the reference genomes are really interesting because they're like literally like there are different releases of the reference genomes, just like software. So like there'll be like some new sequencing method they're able to like better identify certain regions of the genome and they'll update it. They don't do that very often, but there's a new mouse reference genome if anybody cares. I know a lot of people are excited about that. At least in not me, but other people that I work with are. You had a question?
Just an abstract question. Like, so how do they deal with like the sex chromosomes?
Yeah. So they have the X chromosome in there, and I believe they have the Y chromosome in there, but there are also like, there are regions that are paralogous across the sex chromosomes where, but there are also regions that are like unique to the X chromosome with compared to the Y chromosome, but they'll basically be separated by a chromosome there. There's definitely like some chromosomes that are the paralogous regions that always show up and like screw things up, but I can't remember what the exact nature of those are. Like the chromosomes that start or end with PARY, I don't know. Nobody else has looked at the genomes as much as me, apparently. Yeah, they have ways of dealing with it though. And that's like one of the ways you can actually figure out the sex of your sample is you can see like, are there a lot of reads that map to, you know, an X linked or something on the X chromosome, or are there twice as many reads that map to something on the X chromosome as a different sample, which might be a male sample? Are there any reads that map to the Y chromosome? Something like that. Yeah. Okay. So this particular chromosome is actually from a spike-in data set because I wanted to be able to show you the full sequence. Yeah, so FASTQ format has 4 lines and each entry is a read. It's not like a reference sequence. There's no indication of like what that sequence might be or where it comes from. So this is like off of the sequencer. The other difference is that the header lines start with @ instead of the caret, and they have this placeholder and they have the quality score. And this is compared to the FASTA file where you just have the chromosome name and the sequence. So 2 lines for each chromosome in the FASTA, 4 lines for each read in the FASTQ. Yeah.
Maybe more theoretical.
Sure.
Experiment minded. If you're doing single cell RNA-seq, right, and you have your reference genome, can you choose a cell specific one?
So the reference genome is the same across all cells, unless you're working in cancer, which is a very big if. So remember that the genome is the same in all of your cells, and it's like the combination of like epigenetic state and transcriptome that defines your cell identity. So typically, I mean, the genome you can map like any data to. No, it doesn't depend on your cell states. You can fine tune your reference genome. Say you're looking for a specific gene that you know is on like chromosome 21. If you really wanted to, you could like cut everything out of your genome except for chromosome 21 and then just map to that. But I don't really think that there's a huge reason to do something like that otherwise. That's a good question though.
I was thinking hypothetically, if you have a disease that has progression, and if you don't know the basis of it, like you know the gene, but you don't know the cascade down through progression, if you did have something more, say, Huntington's, for instance, MSNs are the primary neuron in which Huntington's disease targets. We don't know why, but if for some reason, starting at the first differentiation age down through the age of the MSN, if it was more prone to having mutations throughout its aging, then if you didn't have a starting cell or an ending point in your reference, you wouldn't know if mutations are more prone to occur throughout a timeline in that cell. Like, that's what I was asking. Like,
Yeah.
the genome is supposed to be the same.
Yeah.
But if it,
No, that's a great question. So you can use the reference genome to do things other than RNA sequencing mapping. So what a lot of people do in that sort of field is they do variant calling. So you'll sequence the genome instead of the transcriptome. So you'll sequence DNA instead of RNA. And then you'll map it against the reference with a specific focus on what nucleotides are different compared to the reference or compared to a reference set of variants. So we have these other files called VCF files, which have basically like a list of like very, very common variants, you know, what makes all of us different from one another, the things that have been profiled. And you could call what's different compared to the reference genome and what's different compared to normal variants. So that's another way that is another way you can harness the reference genome, but it would be possible. Yeah. Any other questions or comments? Okay, so I've been talking a lot about mapping without actually going over what mapping is. So again, this is a very, very high level overview of what's going on when you run STAR or when you run Bowtie, I think. Nobody uses Bowtie anymore. So if you have your reads in FASTQ format, oops, on the left here in dark gray, and you have your reference genome on the right here in light gray, what you want to do is you want to figure out where each read came from in the reference genome. And so the first step to this is compare each read to each location in the genome. And you can imagine that this takes a long time, but I promise that they found ways to really, really speed it up. So you basically take each read and you like check at each position in the genome, like, does it match, does it match, does it match? And the way that they actually compute whether it matches or not is they compute a score for how well each read matches to a specific region of the reference genome. So if we zoom in on these 3 reads here, we can see how they compare to these 3 regions in the reference genome, or I guess this one region in the reference genome. And the way that this score is computed is that perfect matches, which I've highlighted in green here for your convenience, actually increase the score of how well that read matches to the reference genome. Whereas mismatches, insertions, and deletions that are in the read compared to the reference genome will decrease the score. And so again, there's a lot of like algorithmic work that goes into this under the hood, but in essence, what you get out of read mapping is the region in the reference genome with the highest score for each read. And this is usually output in a BAM file, but we're not going to look at any of those today, so I'm not going to tell you what those look like. Okay, any questions here? Yeah, it's very similar to blast alignment. They use a lot of the same principles.
Can you like
Yeah. So it depends on what you're doing. A lot of people in like phylogenetics will do that. So they'll try to see, like, for example, if you have like an organism that you're interested in like seeing what it's most closely related to in terms of other animals that you have reference genomes for, you can map to multiple reference genomes to see basically what organism it matches closely with, most closely with.
I mean, like multiple reference genomes, like of the same organism.
Oh,
Like to increase your, is that like a common practice?
I don't think that people do that. I think that typically the mapping algorithms are really good such that common variants will not disrupt mapping rates. And if they do, you can use like variant aware mapping, or like you can use, if you're working with like a cell line that's really well characterized, it might actually have its own reference genome. For example, I know that the GM12878 cell line actually has its full genome sequence, and it's a separate thing from like the HG38 reference genome. So you could map to that. But again, that's kind of getting into the idea of like genetic variants. So those people do specific things to do that, but I don't think that those individual mismatches usually penalize these things strongly enough to disrupt mapping rates. Good question, though. Do you have a question? Yeah, that's a great question. So that's what I'm going to talk about next. So if we have no other questions, I'll go into that. Okay, so as Tao was saying, if we're sequencing RNA, which is typically messenger RNA, we are looking at spliced versions of these genes. So for those that may not know, if you have this gene structure here in the reference genome, where each of these colored blocks are different exons, and each of these lines are different introns, you can get these two different splice isoforms out of the gene itself, one which contains the red and green exon, and one which excludes them. And so two things you'll notice here are, first of all, that there are no introns in these reads, and maybe that was the only thing. One thing you'll notice is that there are no introns in these reads. So if you're sequencing things that come from these isoforms, which these are indeed the molecules you'll be sequencing in a typical RNA-seq data set, you're going to be getting data mostly that excludes these introns. So in traditional reference genome-based mapping, it does make it a little difficult, as these will be kind of registered as large-scale deletions in your mRNA reads as compared to the reference genome. Although there are mapping strategies that really work well with spliced RNA. But I do want to use this kind of as a segue to talk about transcriptome mapping as well. So for instance, if we have this read here, which it comes from this bottom isoform here, which contains this dark blue and red exon-exon junction. If you try to map it to the genome, it'll be like, I don't really match here, I can only match the blue. And then it'll be like, I can only match the red over here. So what is it going to do? And again, enter reference transcriptome mapping. So this is an additional type of reference file that you have in FASTA format, which actually contains the nucleotide sequence for each known spliced mRNA isoform. So these are nucleotide sequences that contain the isoforms without the introns in them. All the known sequences without the introns in them. And this enables quantification on the isoform level as opposed to the gene level, although that is a dubious claim as somebody who works with long-read RNA-seq data. But it also allows for more efficient mapping of mRNA reads, which again typically come from these spliced transcripts. So if we're trying to map our read now to the transcriptome instead of the genome, it'll be really happy because now it can match both of these exons here. And below, you're just seeing an example of one specific reference sequence that comes from the reference transcriptome. Now in the first line, there's a little bit more information than just the chromosome name. It's actually information about the transcript. So here you can see the transcript ID, the gene ID, the transcript name and the gene name, as well as the transcript biotype. So this is a short RNA, so it's actually a pretty short sequence. Yeah.
So you just explained why we would map mRNA to the transcriptome, right, as opposed to the genome.
Mm-hmm.
So my question is, you're still going to have some, not a lot, but some mRNA that still have introns.
Yep.
Right?
Mm-hmm.
What do you do with those?
There are a lot of things you could do with those. But I'll say one specific example on the next slide, if that's what you're getting at.
Do you need to, like, those like stars, those sort of events, do they know to treat those guys differently somehow, or how does it map?
I'm going to assume that they do, because the people that developed those tools are much smarter than me. So in all honesty, these things are probably not huge problems, but I just want to kind of highlight that there are multiple options for mapping. So if you're not interested in introns in any way, go with the reference transcriptome. If you are interested in introns, go with the reference genome. And Emmanuel, there are ways that you can like tune Callisto arguments to run it in the like RNA velocity mode or whatever, which I'm going to talk about right now. So just again, a brief pros list for mapping to the genome versus the transcriptome. As I just mentioned, the genome allows you to detect introns, which is not particularly useful in any capacity that like a normal analysis pipeline would use, except for if you're going to do RNA velocity analysis. So if that doesn't mean anything to you, that's fine, but just know that you need intronically mapped reads in order to do RNA velocity analysis. For basically pretty much anything else, you probably want to map to the transcriptome. It's most commonly used in these single cell RNA-seq studies. Yeah.
So if you have a single nucleus data set, you actually need to provide a pre-mRNA reference, which includes it. Because in the nucleus, you're going to have more on the splice with when the mRNA is. So you can either supply a different reference. And some softwares these days are smarter, and you just tell it to include introns, and then it's like, okay, so, but yeah, you do have to account for it somehow.
Yeah, okay, that's a really good comment, and I'm glad you brought it up.
Especially for single nucleus.
Yes, so as a brief biology reminder, splicing occurs in the nucleus. So if you're sequencing nuclei as opposed to whole cells, you will be getting a lot more of unprocessed messenger RNA from the nucleus. So this does necessitate, if you want to get like a higher mapping rate, using the introns for that. So thank you for bringing that up, and I will edit these slides after that to put that in there. Yeah, what's up? All annotated splice variants, which is a subject that's up for debate if it's all of them, because we don't know a lot of them, in my opinion. But yeah, they do their best.
Back to the so if you're doing sequencing, then you're saying that you have to, for mapping, you're saying like your reads will have intronic nucleotides in them, right? That's what's going on. And then, so then we need to map that. It's better to map that to the genome.
Do you want to answer that? Because it sounds like you know much better.
It's not genome, it's actually something called like pre-mRNA. Oh, okay. So it's going to be the sequence, but instead of the splice version, it'll be the version that includes the introns.
So it still won't contain like regions that are in between genes like the genome would,
right,
but it will contain the intronic regions for that transcriptome as well.
Okay, yeah. So it'd be like another column on this, but like pre-mRNA.
Yeah, I guess so. Yeah.
Yeah, and a lot of times you can just like wherever you would normally find your reference transcriptome to download, you'll see like, oh, there's the pre-mRNA. Gotcha. Yeah. Thank you.
Yeah.
I have a follow-up question. So if you do complete nuclear, is there an option where you can look at both pre-mRNA and complexing RNA? I think you could have to map them separately. Okay. There's no like combined reference. I mean, you could. Oh, there might be, but you could definitely combine them yourself. But then there might be the problem where something is like mapping highly to two regions. And I don't know how it resolves that. Okay.
Yeah. I think I mean, typically like for, if you're doing RNA velocity analysis, you'll get like your spliced RNA matrix and your unspliced RNA matrix. Your unspliced ones will be reads, like quantification from reads that contained introns, and your spliced one will be from RNA reads that did not contain introns. And is that done by mapping both to the genome or pre-mRNA transcriptome and to the spliced mRNA transcriptome? Okay, never mind. Okay.
It's like if it doesn't matter, there's a way that it tells if there's any introns in the sequence.
Okay.
And if there's not any introns, then it's okay. This one's been spliced.
Do you provided the GTF?
Yeah.
Okay, that's why. Okay, cool. Good to know. All right. Any more questions on this? These are good things to be thinking about, although it's going to be like a blip in your analysis, hopefully. Okay, I'm almost done. All right, so now we know where each read comes from within the genome or the transcriptome, and we know which cell each read comes from. So the last question is now, how can we actually generate our counts matrix? How can we determine how highly expressed each gene is? And this answer is actually just as simple as taking your genome annotation, which tells you where the boundaries of each gene are in your reference genome, and then counting up all of the reads that mapped to each of those regions. So if you have 4 genes that mapped to gene A and 4 reads that map to gene A and 2 reads that map to gene B, this is what your counts matrix is going to look like. And yes, I'm assuming that these reads have been UMI corrected. Sorry. Good point though. Yeah, okay. So thankfully for you, we did that. Thankfully for you, there's a whole boatload of software tools that already do all of this stuff, where you only have to like tune hyperparameters to it or whatever. That includes Cell Ranger, Split Pipe, Star Solo, Alvin, and Callisto bus tools, as well as many others. And most of these perform cell barcode identification, UMI deduplication, read mapping, and quantification.